Good morning. I'd like to call this meeting to order. My name is Ann Aiken. I'm the acting designated federal officer for the National Vaccine Advisory Committee. There are a few things I want you to know before we get started today. First, this is the public meeting and it's being recorded. All statements made today are on the record. Second, this advisory council is governed by the Federal Advisory Committee Act, or FACA for short. FACA provides rules about the circumstances by which agencies or officers of the federal government can establish or control committees or groups like this one to obtain advice or recommendations. The voting members are special government employees who are therefore subject to conflict of interest laws and regulations, as are all members who work for the federal government. These members previously provided information about their personal, professional, and financial interests. Each voting member's financial interest and outside affiliation has been carefully screened, and this happens every year, to ensure that they comply with federal ethics laws. The liaison representatives are non-voting members of the advisory council and are not subject to the same FACA rules as the voting members. Additionally, the information provided at this meeting does not necessarily represent the official position of the National Vaccine Advisory Committee or the U.S. Department of Health and Human Services. Mention of products, processes, services, manufacturers, companies, or trademarks does not constitute its endorsement or recommendation by the U.S. government, HHS, or NVAC. So I'm going to go ahead and start roll call for today's meeting. If you are on the Zoom, please come to camera and take your mic off. And if you're in the room, please use your microphone. Bob Hopkins.
Here, thank you.
Good morning. Melody Butler.
Present. Good morning.
Melody, I saw you on, but we cannot hear you.
Hello.
Yeah. Good morning, Melody. I hear you now.
Thank you. Pleasure to be here.
Dan Hawk, are you on? Dan, we cannot hear you. Molly Howell.
Present.
Good morning. Jewel Mullen. I also saw her on.
Present.
Oh, morning. Steve Rennernick.
Good morning, present.
Winona Stolzfus. Winona, I saw you on as well.
I'm here, Ann. Sorry, present.
Oh, good morning.
Good morning.
Geeta Swami.
Present. Good morning.
Good morning. Bob Swanson.
Present.
Brenda Holbrook.
Present.
Good morning.
Morning.
Rebecca Coyle.
Present.
Mitch Rothholz.
Present.
Morning. Erica McGowan.
Present.
Good morning.
Good morning.
Judith Schley. Judith is the Associate Director of Public Health Institute at Denver Health and is the new NVAC member representing NACHA.
Present.
Good morning. Aaron Henry.
Present.
Hannah El-Sali. Okay. Justin Mills. Kim Armstrong.
This is Christina Shansky on her behalf. I'm present.
Oh, hi, Christina. Captain Manisha Patel.
Good morning. Present.
Good morning. Mary Beth Hans. Lieutenant Colonel Nicole Sue. She's also a new member representing the
Good morning, present.
Good morning. Uza Chukwuma.
Good morning, present.
Good morning. Andrew Ford.
Good morning, present.
Good morning. And Troy Knighton.
Good morning, Ann.
Good morning. Before I introduce Admiral Levine, I just wanted to recognize our committee chair for his outstanding leadership. I also wanted to thank the team, which includes some people sitting over there, Rebecca Lazaration, Emily Downs, Chloe Loving, and Viola Jacobs. There's also a slew of people running around here helping to escort members in and working at the registration desk. I also wanted to thank the studio team, our tech support, and our language interpreters for their help today. And then I wanted to thank our leadership, both Kay Hayes and Tim Harrison, our office director and principal deputy director, provide consistent and thoughtful leadership to the National Vaccine Program. And I also wanted to thank them for their efforts, as well as Dr. Chinedu Okeke, who is the acting division director for the National Vaccine Program team. He'll be here tomorrow and will present and lead a discussion on some work that they are doing. So it's now time to introduce Admiral Rachel L. Levine. She serves as the 17th Assistant Secretary for Health for the U.S. Department of Health and Human Services and the head of the U.S. Public Health Service Commission Corps. She is also the director of the National Vaccine Program. She fights every day to improve health and the health and well-being of all Americans, and she's working really hard to ensure health equity is at the front and center of all the work that we do to build a stronger foundation for a healthier future, one in which all people and places in our nation can thrive. Admiral Levine has a storied career, first as a pediatrician and adolescent medicine specialist. She worked in academic medicine and focused on the intersection between mental and physical health, treating children, adolescents, and young adults. Then she worked as Pennsylvania's Physician General and later as Pennsylvania's Secretary for Health, where she addressed COVID-19, the opioid crisis, behavioral health, and many other health challenges, as she does today. With that, I'll turn it over to Admiral Levine to provide her opening remarks.
Well, good morning. Thank you, Anne, for that introduction. And greetings, NVAC members, speakers, and others who have joined us in person today, but also across the nation on live stream. I'm really pleased to join you today to provide opening remarks. It's June. June brings warmer weather, especially the next couple of days in Washington, D.C., hopefully sunny days, but also so much pride as we celebrate the LGBTQIA+ community, A for allies. I want to take a moment to recognize the LGBTQI+A community for their commitment and their contribution to public health. So happy Pride Month for everyone. When I see the LGBTQIA+ community, I see hope. I see hope for a more equitable future. I see bravery in each one of us being ourselves, despite the challenges that we continue to face. And I see strength from love, friendship, and community, because we are stronger together. Pride is our time to celebrate our community and reflect on how far we have come, but also how much farther we have to go. Pride reminds us that we are a strong, resilient, and powerful community that fights hate with love. Pride Month always inspires me. The Biden-Harris administration promised to support the LGBTQIA+ community from the very beginning, and I joined that promise to promote health equity for all, including the community. HHS has completed the first year of implementation of its Sexual Orientation and Gender Identity Data Action Plan, or SOGI Data Action Plan, which has advanced data equity within numerous instruments across HHS divisions. We encourage, of course, healthy behavior, which always includes vaccinations. It is part of a syndemic response that aims to increase Mpox vaccine uptake for people most at risk in terms of advancing health equity. The terms syndemic and health equity are important here as Mpox vaccination disparities persist for gay and bisexual men and other men who have sex with men, as well as racial and ethnic minorities, and that intersectionality of health equity. These disparities call for sustained equity-based strategies, such as culturally tailored messaging and data-driven expansion of vaccination services to better reach those most impacted. So this Pride Month, as we did last year, we are strongly promoting Mpox vaccination, awareness messaging and testing, alongside relevant public health services such as HIV and STI testing, especially in this current climate of testing and treatment for syphilis. We also connect people with mental health, as well as harm reduction resources, housing assistance, substance use disorder treatment, and more. So you're going to be hearing more about how we're working all together, stronger together, and to catalyze change for the LGBTQIA plus community later today. Even I get tongue-tied with our rainbow family. Before you participate in the upcoming session and discussion, I do want to take a moment to celebrate a few successes of the Mpox response. First off, studies show 2 doses of Mpox were up to 86% effective in preventing Mpox. Secondly, the vaccine is now widely available through health providers, clinics, and other locations such as Pride events. And I encourage people to use HIV.gov and the locator to find Mpox vaccination that is near to them. Later today, you will hear about the wonderful work to engage the LGBTQIA plus community, to share resources, work with communities to provide tailored and targeted solutions to reduce barrier to vaccination, take advantage of any missed opportunity, and to share resources and knowledge because we are stronger together. In regards to global immunizations, I want to celebrate the recent study findings that were published in The Lancet. The data revealed that global immunization efforts have saved at least 154 million lives, 154 million lives over the past 50 years, 6 lives every minute of every year for 50 years. I have said many times that vaccinations, immunizations are one of the victories of public health in the 20th and 21st centuries. This is a significant and exciting milestone for public health, and I think we should take a moment and during the session tomorrow to recognize this truly amazing accomplishment. And clap down. While gaps remain nationally and globally, we all have been a part of one of the most beneficial public health achievements, vaccinations. A time-honored effort to protect ourselves, our patients, our families, our communities, our nation, and the world from serious diseases. Vaccines are one of the most effective and cost-efficient public health interventions that exist. They have helped us eradicate smallpox, get very close to eradicating polio. They help us fight COVID-19, influenza, Mpox, just to name a few of the of vaccine preventable diseases. In fact, vaccines are a cornerstone to individual public health, family public health, community public health, national public health, and global public health. We have a representative from the WHO speaking at this meeting to share more information about this wonderful accomplishment in a new campaign called Humanly Possible. I love that name, Humanly Possible. It is a joint campaign from UNICEF, WHO, Gavi, and the Vaccine Alliance, the Bill and Melinda Gates Foundation, and others to affirm their commitment to vaccination and immunization services. You will also hear from speakers today and tomorrow about the dangers of resurgence of vaccine preventable diseases, including a recent outbreak of measles in Illinois. We had no cases of measles in the United States in the year 2000. And here we are with outbreaks of measles. Measles was declared eliminated from the United States in 2000, but we can and still do see cases despite a very safe and effective vaccine, which can prevent 93% of cases after 1 dose and 97% of cases after 2 doses. Outbreaks like this serve as a reminder of the importance of vaccinations, the importance of our public health response, and an optimized vaccination system in our country. But it also is important to highlight, as I talk with Director Mandy Cohen of the CDC, that we still do have very good immunization rates and very good measles immunization rates. So we don't want to state that it's a crisis at this point in terms of the measles vaccination rates. However, certain states, certain communities, certain areas of our country, and certainly around the world are lagging in terms of the immunization rates, and we have to double and triple our efforts. So with this in mind, I am charging this committee with a new task. I would like your insights on ways we can continue to make significant progress against vaccine preventable diseases in the United States. and better address the persistent and significant gaps in vaccination coverage that contribute to preventable morbidity and mortality in our country every year. As we've done in the past, HHS will release an updated Vaccines National Strategic Plan. The team, led by Kay Hayes in our Office of Infectious Disease and HIV/AIDS Policy, has started planning to update the 5-year national strategy in 2026. We are already planning for 2026. You will hear more about these efforts tomorrow, and I really hope that you will provide us with your initial insights for the change during the discussion tomorrow. This current plan was developed, much of it was developed before the lessons of COVID-19 and the Mpox response. So I'm interested to hear your thoughts on vaccine readiness and ways that we can integrate our new information and new things that we have learned into the plan. Additionally, it's important to remain responsive to the evolving vaccination landscape. And I would like to hear your thoughts about pressing systemic issues such as vaccine hesitancy, equitable access to all routinely recommended vaccines for children, adolescents, young adults, and even seniors like me. As you know, NVAC has a long history of providing HHS with guidance in plan development and last approved a report in 2019 that was used in the development of the current plan. So, with gratitude for your previous work, your current work, and your future work, I charge NVAC with the following tasks. Review and assess the goals, objectives, and indicators of the current Vaccine National Strategic Plan 2021-2025. And some of this will be done tomorrow, but we need a deep dive on this. Seek input from a diverse set of experts in the development of the report recommendations and guidance, and Anne can help you secure additional expertise as needed to respond to this charge. Thank you, Anne. Keep or propose new goals, as well as up to 10 new indicators that are measurable to help determine if the goals and objectives of the plan are achieving their desired outcomes. Measuring outcomes is vital and essential to our success, and you'll be discussing that tomorrow. Recommend and prioritize 3 objectives within each of the 5 goals that the committee thinks are poised to make the greatest impact in the next 5 years. This is another important component of the charge, and we look forward to hearing your recommendations. And write a short report to be voted on at the February 2025 NVAC meeting documenting this guidance. So I look forward to reviewing your recommendations and applying them to all our work moving forward. So in closing, thank you so much for your time today. I, as always, appreciate your dedication and your commitment and your expertise and your experience. I could keep going to optimizing NVAC's work. I look forward to working with you and want to know, and I want you to know that all of that, what I just said, your expertise, your experience, your work is invaluable to HHS, invaluable to me as we strive to improve the health of our Americans. So thank you so much. And now, as always, I will turn to Dr. Hopkins to provide today's welcome. Thank you.
Well, good morning. Thank you for joining us for today's NVAC meeting. Thank you, Dr. Levine, for your thoughtful remarks and for the new charge. We look forward to the discussion today and the work moving forward. And thank you, Anne, for getting us started, and to the NVAC team for its work in planning this meeting and supporting the committee throughout the year. I also want to thank the NVAC members and the subcommittee members for their dedication to further the work of this committee. I look forward to the presentations and discussions today and tomorrow. I want to begin with a few housekeeping items, and we can move on then to the review and approval of the minutes from the February meeting, followed by a high-level overview of the agenda. In terms of housekeeping, I want to make sure everyone is aware that this is a public meeting, and it's being webcasted on the HHS website. I want to let everyone know that we have sign language interpreters available on the HHS live stream. We ask that everyone speak slowly and clearly and assist them in supporting our meeting. For our virtual participants, please remember to mute yourself when not speaking, and please do not use your camera unless you're presenting, asking questions, or answering a question. During discussion, I ask that all members and speakers, virtual and in person, identify themselves before speaking if I didn't acknowledge you by name in giving you the floor. This helps the note taker and others to follow along. Throughout the day, there will be opportunities for committee discussion. If you'd like to ask a question or provide a comment, send me a message through the chat feature if you're not here in the room. The next stage, I would like to proceed to approval of our meeting minutes. NVAC members were previously sent the minutes from the February 23-24 meeting. Do any of the members want to make corrections to those February meeting minutes? Hearing none, could I get a motion and a second for approval of those minutes?
Motion to approve.
Thank you, Dr. Swami.
Second.
Second from Molly Howell. Since we are hybrid, I will ask that anyone that has objection or does not vote approval to make a notice, please. Don't see anybody in the room. Ann, are there any online? All right, we will consider our minutes approved from the February meeting. As always, members of the public can provide a public comment by phone at about 4:15 p.m. today and at 2:15 p.m. tomorrow. Public comments are not a question and answer session. They represent an opportunity for individuals who'd like to make a statement to do so. The deadline to request a space for public comment during this meeting has passed. However, anyone can submit a written comment up to 3 pages in length to NVAC@HHS.gov. Spend a couple of moments talking about the highlights of our meeting that we have planned. We have a wonderful lineup of speakers. Today, we'll start with a panel on proactive production capabilities for rapid response to emerging diseases. Following a brief break, we'll have a panel on equity for Mpox vaccination as it relates to pride and to community. After lunch, we'll hear from experts about the upcoming fall and winter respiratory disease season. We'll then hear an update about an outbreak of measles in Illinois. And finally, we'll hear about breast cancer vaccine innovations. We'll close the day with public comments. Tomorrow, we'll open with a panel on innovations and improvements in immunization data. Following a short break, we will hear an update on global immunization efforts and how vaccines have saved over 154 million lives worldwide. We'll then hear about research on switching arms between COVID-19 vaccine doses. Just before breaking for lunch, we'll hear updates from our federal agency and LASSON members. After lunch, we'll hear from experts from the U.S. Department of Health and Human Services about their work toward an updated national vaccine strategy. And the day will again conclude with public comments. Sorry, I got ahead of myself. Finally, as a reminder, please hold our remaining 2024 meeting dates on your calendars, September 12th and 13th of 2024. Please refer to the NVAC website for final details on upcoming meetings. At this time, let's turn to our first panel. Our first panel is entitled Being Ready for Rapid Response, a proactive discussion of production capabilities. While the current public health risk of novel influenza in humans is low, it's incredibly important for public health officials to continue to monitor bird flu viruses and any changes in their ability to infect people or other animals. In this session, we'll explore readiness capabilities to produce safe and effective novel flu vaccines. Our speakers for the session include Dr. Christine Oshansky, who's the Influenza and Emergency Infectious Diseases, Pandemic Vaccines and Adjuvants Program Chief in the Administration for Strategic Preparedness and Response at HHS. Dr. Lorna Meldrum, who's the Vice President of Commercial Operations for the International Markets and Pandemic at CSL Seqirus. Julian Ritchie, who's the Vice President and Head of Vaccines and Public Affairs and Patient Advocacy at Sanofi Vaccines. Dr. Oshansky, your slides are up.
Great, thank you.
I
Thank
see you
you.
on
Can you hear
mic.
me okay?
You are loud and clear. You have the floor. Thank you.
Great, thank you so much. So my name is Christine Oshansky. I'm the Program Director for the Pandemic Vaccines and Adjuvants Program in the Influenza and Emerging Infectious Diseases Division at BARDA, the Biomedical Advanced Research and Development Authority. You can go to the next slide. So we're part of the Administration for Strategic Preparedness and Response, or the ASPR, and the ASPR's mission is to assist the country in preparing for, responding to, and recovering from public health emergencies and disasters, and that includes pandemic influenza preparedness and response. Next slide. And BARDA sits within ASPR. So BARDA, we develop and make available medical countermeasures by forming unique public-private partnerships to drive innovation off the bench to the patient to save lives. And we do this, and we have various ways of doing this. We have flexible, nimble authorities. We have multi-year funding. We have cutting-edge expertise in-house, as well as facilitating partnerships with our vaccine manufacturer partners that you'll hear from today. And as such, we promote innovation. Next slide. So our pandemic preparedness policy is outlined in many different preparedness plans. Some of them are listed here. And the US government has established several goals within these plans. And the goals include maintaining enough antigen and adjuvant to rapidly formulate and deploy vaccine to vaccinate the highest priority population groups in the US. It also includes a goal of delivering first finished doses of vaccine within 3 months of a pandemic declaration, meeting public demand within 4 months of a declaration, and then having a goal of manufacturing, fill finishing, releasing, and delivering enough adjuvanted vaccine for the US population shortly thereafter. And so all of these goals align with the BARDA strategic plans, goals 1, 2, and 3, preparedness, response, and partnerships. Next slide. So if we go back in time, around 2005, there were only 2 US licensed seasonal influenza vaccines, 1 US-based manufacturer. Only 1 technology was used in the manufacturing process. That was egg-based manufacturing. Only 1 formulation was used, so that was a 15 microgram per dose in a trivalent form, and there was really no pandemic vaccine data available. BARDA became BARDA in 2006, and if we fast forward through the 2009 H1N1 pandemic, and then moving into 2017 through 2019, where we were preparing to possibly respond to an H7N9 outbreak due to the epidemic that was seen in Asia. By then, we have multiple licensed seasonal influenza vaccines, 3 US-based manufacturers, 3 different technologies. So not only the egg-based, but moving into the cell-based technology, as well as the recombinant protein-based manufacturing technologies. We have multiple seasonal formulations. And by this time, we have FDA licensed pandemic vaccines, and we have our national pre-pandemic influenza vaccine stockpile program in place. And then if you look to today, we've come out of responding to COVID-19, of course, and we have some very bold goals that are outlined in the American Pandemic Preparedness Plan, or the AP3. And so this is to get to first dose of vaccine within 100 days of sequence availability, getting to meet the US population needs within 120 or 130 days, and that all leads to faster distribution and administration. And we have several initiatives in place at BARDA to try to meet these goals. And so that includes developing faster vaccine platforms, and our mRNA-based vaccine is a good example of this, looking into alternative delivery technologies, looking into single-dose formulations. How can we broadly prime using a vaccine, especially in the context of pandemic influenza? And all of this is thought about in the context of US-based manufacturing capacity for these new technologies. Next slide. So we do have available medical countermeasures. So for pandemic influenza vaccines, we have CSL Seqirus' Audens. We have GSK H5N1 vaccine and Sanofi's H5N1 vaccine. And we, of course, have seasonal influenza vaccines that we see every year. There are also outpatient treatments that are listed here, like oseltamivir and baloxavir, zanamivir, and peramivir. But while we do have these medical countermeasures, gaps do exist. So these 3 licensed pandemic influenza vaccines here in the US, they're H5 specific. And so the gap includes expanding those vaccine licensures to other subtypes like H7 or H9 or H2. Having vaccines that are better and faster, can we manufacture them even faster than they already can? Can we get to the greater immunogenicity faster? And then all of this is, the focus is on the safety and immunogenicity data so that we can possibly reduce response timelines. Next slide. So here in the influenza and emerging infectious Infectious Diseases Division, our goal is to establish new and leveraging existing infrastructures and capabilities to support response and preparedness. So we do this via flexible agreements. So you'll hear a little bit about that with our partner, that we have agreements with our partners like CSL Seqirus and Sanofi and GSK. We're always looking for platforms for faster development. And we do all of this in the lens, not only of safety to the patient, but also in the context of sustainable manufacturing, because we can do nothing if it's not sustainable. Next slide. So our program, the Pandemic Vaccines and Adjuvants Program, our main goal is to advance timely vaccination strategies to mitigate the next pandemic. So on one hand, we work to accelerate pandemic response timelines. So we're always looking to identify areas for improvement to the licensed manufacturing processes. And we do this in close collaboration with our partners at CSL Seqirus, GSK, and Sanofi. Through these partnerships, we also manage the U.S. National Pre-Pandemic Influenza Vaccine Stockpile. And this program contains a robust library of influenza virus vaccine seeds, as well as bulk antigen lots, so that the manufacturers can move into rapid production as quickly as possible if the need arises. And because we have these partnerships already in place, it allows us to have expedited procurement of vaccine if needed. On the other hand, we work very hard to ensure that clinical and analytical data exists to support vaccine administration. So not only do we look to have nimble vaccine implementation strategies in place, so one example of that I'll talk about in a little while is the heterologous prime and boost antigen strategy, for example. But we also want to expand safety and immunogenicity data sets for influenza vaccines targeted to diverse subtypes and diverse strains. So not just H5 and H7, but H9 and H2. So those are some of the gaps that we're looking to fill. I mentioned this already, but we look to generate data to support administration. So this includes physical and chemical compatibility studies, vial sterility assurance studies, extractable volume studies, just to make sure that when the vaccine does arrive in the clinic or at the administration site, that all the data exists for that administration to occur safely. And we also look for, perform proactive assessments to confirm that the antigen and adjuvants that are in the inventory remain safe and immunogenic over time. Next slide, please. So how do we make decisions about pre-pandemic influenza vaccines? So BARDA is part of an interagency decision-making body that includes our sister agencies within HHS like CDC and FDA and NIH, but also other agencies in the US government like the VA and Department of Homeland Security and others. And we come together quite frequently to examine the surveillance and epidemiological data, not just on the seasonal influenza virus side, but also the zoonotic influenza viruses that may be circulating in wild animals across the globe. And using this data, we make decisions using a metered response approach. And so one example of this is CDC's Influenza Risk Assessment Tool, or their IRAT, where each of the strains is scored based on their relative, the risk of impact to public health and based on the risk of emergence. So each of the strains are scored, decisions are made, and then those decisions are implemented based on timing and funding availability. And so on the right is just a notional graph of some of the decisions that could be made. Maybe nothing needs to happen because we already have a seed lot manufactured. Sometimes we'll make antigen bulk lots, perhaps a clinical trial will be run, and all this is in the context of pandemic preparedness. Next slide. So this next slide is just an overview of what I've already said. The CDC monitors disease outbreaks, looking at surveillance across the surveillance in wild animals and as well as in humans. Risk assessments are performed. Now, concurrently, the WHO collaborating centers are generating candidate vaccine viruses to make sure that the manufacturers have those available if needed. And likewise, the WHO essential regulatory laboratories are making sure that potency reagents are ready to go if needed. And then our partnerships with CSL Seqirus, GSK, and Sanofi, we work with them very closely to develop those seed lots, to manufacture bulk antigen, get those into final containers if we need. And anything that is manufactured gets placed into stability programs. So they're always monitored at the manufacturing sites so that they remain within specification. Clinical trials are sometimes performed, and any data that is generated gets placed into pre-emergency use authorization living documents. So these are living documents, they're updated periodically, and they're on file with the FDA so that we can rapidly respond if we need to go. And with all of these, all of this data in place, we strive for it to lead to faster to first doses and faster to full immunization if the need arises. Next slide. So these are just a few snapshots of some of the clinical trials that we have going on. We're very focused on generating safety and immunogenicity data. I'm going to go through these kind of fast, but all of this is public data. It's either published in the literature or it's on clinicaltrials.gov. So the first study I just want to highlight is this was a study looking at Sanofi's H7 recombinant protein antigen that was adjuvanted with either AS03 or MF59. And you can see really nice antibody responses that are sustained even out to 6 months. Next slide. This study is a more recent study from GSK. They were looking at their H7N9 influenza virus vaccine candidate using their Fluvaval process. This study looked at varying doses of HA adjuvanted with AS03, either full dose or half dose. And the idea here was, can we get more adjuvant doses out of one lot of adjuvant if we need to, if we're adjuvant constrained, for example. So we wanted the data in hand. So there is an error on the slide. So these are not seroprotection rates. These are seropositivity rates. But the bottom line is either in adults 18 to 64 or adults 65 years of age or older, the seropositivity rate after 2 doses is relatively high across the board. Next slide. This is a study that was performed several years ago. This is a study outlining or highlighting that the antigens that are in the stockpile as bulk material remain safe and immunogenic. So this was a study looking at Sanofi's Vietnam 1203-2004 vaccine administered with and without adjuvant. As you can see here as well, that the vaccine is safe and immunogenic when administered as a 2-dose regimen, even when it's stored for long periods of time. Next slide. This is the heterologous prime and boost study I was mentioning. So this is a BARDA-sponsored trial looking at varying antigens that we did have represented in the stockpile, and the manufacturing processes are listed here. And this was across varying H5 clades that have been circulating since 2004, 2005. And so what we did here is we administered 2 doses in a heterologous prime and boost manner, 21 days apart. And then in a couple of the groups, we gave a 3rd dose 4 months later. And on the right are seroprotection rates with the 95% confidence intervals, looking at the different prime boost regimens. So you can see here that it doesn't seem to really matter the order in which you give the vaccine. All of the groups, at least the top 4 bars, they hover around 50% seroprotection rate observed. And then in the groups that received 3 doses of vaccine, you get that nice boosting effect 4 months later. Next slide. So some of these studies are still ongoing, but I did just want to highlight them. So CSL Seqirus is running a study on H2 vaccine. So we know H2 hasn't circulated since the 1960s, and those individuals born after 1968 have never seen H2. And so we are proactively, we're leaning forward to make sure that we have data that exists for a more recent H2 strain that is circulating in chickens. That pops up every once in a while. And so this study is ongoing. Next slide. And then relevant to the current H5 outbreak in wild wild birds and in poultry, as well as the dairy cattle. GSK and CSL Seqirus are both running clinical trials testing their H5N8-like candidates using the Flulavol process at GSK and using Auden's process at CSL Seqirus. And both of these studies are ongoing, but enrollment is complete. Next slide. So last, I just want to touch on the influenza vaccines research and development portfolio. So we work very closely with the vaccine development program in our division, as well as on the CBRN side. But we have several initiatives in the advanced vaccine development space. So we're always looking for faster vaccine platforms and looking for next generation influenza vaccine to enable the manufacture of safe and effective vaccines against novel influenza viruses to get to that 100-day goal. We also look for faster vaccine delivery mechanisms, always looking for greater efficacy and improved vaccine performance. And as always, all this is done in the lens of manufacturing capacity. So we need to enable the production of enough vaccine for the United States population. And so we don't have the manufacturing capacity for these products, then we won't meet that goal. And I believe this is my last slide. Next slide. Yes, so thank you very much. I'll pause here and turn it over to my colleagues at CSL Seqirus and Sanofi.
Thank you, Dr. Oshansky. Our next presenter is Dr. Lorna Meldrum from CSL Seqirus.
Hello, everyone. Can you hear me?
You are loud and clear. Your slides are up. Thank you.
Thank you very much. My name is Lorna Meldrum. I'm here representing CSL Seqirus. I head up international commercial, but also the global pandemic response team. We would very much like to thank the committee for inviting CSL Seqirus to present today. Could I have my next slide, please? So just who are we? Who are CSL Seqirus? We were formed as part of the Australian government over 100 years ago. And one of the first challenges the organisation had to deal with was Spanish flu. And so we've really been involved in influenza for a very long time, over 100 years. We're the second largest producer of influenza vaccines in the world. And at the moment, we hold pandemic preparedness agreements with over 30 governments worldwide. We have an extensive manufacturing network, both our primary manufacturing sites on 3 continents, but also our fill finish capabilities in many other countries around the world. In 2009 and 10, the H1N1 pandemic, out of 2 of our facilities, we responded within 100 days. 100 days from the declaration of pandemic to having a registered vaccine. We have 3 registered vaccines worldwide. We have the cell-based technology adjuvanted with MF59 out of the Holly Springs facility in North Carolina. We have egg-based technology in Liverpool, which is also adjuvanted with MF59. And then we have egg-based technology in Parkville in Australia, adjuvanted with alum. During the COVID-19 pandemic, at the behest of the Australian government, we partnered with AstraZeneca and we manufactured 50 million doses of the recombinant vaccine, the AstraZeneca vaccine, and that was used in Australia and also 5 other countries in the Asia-Pac region. We have in development a COVID seasonal vaccine on our new platform, sa-mRNA. That vaccine has now been registered in Japan, and we plan to make it available in the Northern Hemisphere 24 season coming up. And we are seeking approvals for that vaccine in the EU, in the US, and in other international countries. We have a vast R&D program ongoing with our adjuvanted cell platform, both in seasonal and pandemic, and also with the new technology, our sa-mRNA vaccine technology. Next slide, please. One of the areas that we are really proud of at CSL Seqirus is our partnership with BARDA at Holly Springs. This has been a very long-term partnership, 17 years, gone through several owners, but we have been responsible for that partnership for the past 8 years. The partnership began when the US wanted to move away from its dependence on egg technology and was interested in new technology. So the objective for Holly Springs was to provide cell-based vaccine and the adjuvant manufacturing capacity to be able to deliver 150 million doses of the vaccine in the event of a pandemic. And from the factory being built, or the ground broken in 2006, we're really proud that in May 2022, we were declared pandemic ready. We have been awarded 7 contracts to date by BARDA. The first and foremost was the manufacturing infrastructure, working together to build the facility at Holly Springs, and then subsequently adding to that the viral pilot plant and the small-scale filling facilities. Another really important thing was some contract awarded to improve the yield of the vaccine. If you think that egg technology has been developed over 70 years, so 70 years of year-on-year improvements on yield, whereas this is a new technology. So we had a process called 3.0 that really drove the yield from the facility. When we first took over the facility, the yield for the seasonal vaccine was sitting at about 2 million, and now we can produce 60 to 70 million doses of seasonal vaccine. In addition, as Christine highlighted, we have clinical programs obviously running in seasonal, but in pandemic. And at the moment, we have those studies ongoing for H2N3 and for H5N8. And again, in terms of stockpiles, we work very closely with BARDA to work out which strains they feel are the most relevant for us to produce. So we produce annual pre-pandemic antigen lots, and we also have bulk antigen ready in case of a pandemic or an outbreak need. Next slide, please. So maybe I can say a little bit more about our technology. So we have our cell culture technology at Holly Springs. Because we don't use egg, we can show a greater match to the circulating strains, which leads to greater effectiveness. It also reduces the dependence on egg-based vaccines. If there's a worry about the flocks of chickens that are needed to produce vaccine. Then we have the MF59 adjuvant, dose sparing to the antigen, also adding breadth of protection across different strains. But also because this adjuvant is used in our seasonal vaccine worldwide, we have a very extensive database. To date, over 300 million doses have gone into arms. And then we look to the future, and this is very much potential, but what we want to show is higher effectiveness, longer durability, which is one of the issues with the current mRNA vaccines, multi-pathogen, and of course, speed. And what I hope you see from this is that we're very, very aligned to the areas that BARDA is looking at. So from these 3 technologies, what we are able to offer governments is advanced purchase agreements to the vaccines, either in syringes or vials, pre-pandemic stockpiles for outbreak resilience, and we'll come back to that in a couple of slides, And we have very special technology partnerships, I would say. Obviously, we've highlighted the BARDA partnership, but we also have partnerships with the Australian government, the UK government, and recently, as HERA has been formed in Europe, a partnership agreement with HERA in Europe. Next slide, please. So with this sort of diagram, this cartoon, I wanted to try and explain how we look at what needs to be ready to really be able to deliver to either an outbreak or an epidemic or pandemic readiness. Obviously, you need expertise, both in the receiving governments, but also within our company. Surveillance, and Christine outlined it, we are also very much watching the surveillance, working with the WHO, working with our partners to find out which of those strains are causing them most worry and where they want to be prepared. It's also really important that we have those industry and government partnership agreements signed, sealed and delivered before there is any pandemic or epidemic. It means that we really know what our role and our obligations are, and we can quickly work to deliver that rather than being caught up in what could be detailed and long negotiations. For both of these, we need regular discussions and meetings with our partner governments. That really helps us to respond and meet their needs as outbreaks or pandemics start to form. The framework, again, needs to be agreed in terms of R&D. So we obviously have our view on what R&D we would like to be doing, should be doing, are doing, but that needs to be very much in partnership. What clinical studies need to be done? What should those studies look like? How should they be designed? And again, as mentioned by Christine, again, the regulatory environment, knowing what the regulatory bodies are around the globe, what they require in a pandemic, what can be accelerated. And then the onus is on us within the company to make sure our regulatory dossiers are as up to date and as complete as can possibly be, so that we can once the results are in, we can immediately submit our dossiers. Then we come down to operational status and warm base. And this is something that's critically important to making pandemic vaccines. So our manufacturing operations, obviously advanced manufacturing, it's really critical that the machinery, all the data systems are working, that the people who are using them know what to do. So because we're doing it on a seasonal basis, we have that machinery, that expertise all ready to go. We have staff trained, we have all our quality documents, all our SOPs. So we are absolutely pandemic ready. That also flows into the supply chain. Obviously, we need to hold everything to respond to our obligation to the US population and the US government. So we need to ensure that we stockpile everything to be able to deliver those 150 million doses. Now, some things it's quite clear in terms of chemicals and cells, but we also need to make sure we've got the right number of vials, stoppers, bits of tubing, filters that are ubiquitous, but in a pandemic actually become short-stocked. So all of that we need to plan for. And we also need to plan for packaging in terms of shipping those vaccines out. So that's something we take very, very seriously and is absolutely central to what we can provide to governments. And then moving on to seasonal infrastructure. So if I start with distribution channels, having a really robust seasonal program is absolutely critical, because then the distribution channels are there to take the vaccine from our facilities through warehousing into either pharmacies, physician clinics, offices. That whole supply chain and distribution is there. And then we come to the vaccination rates. It's really important that both physicians and patients are confident about the vaccines going into their arms. And we see that when you have a really robust and successful and high rates of seasonal influenza vaccine, that will lead to a very good response rate during the pandemic. And what we have seen globally, and what I know we've seen in the US, is we've seen a falling off of those seasonal vaccination rates from the very high rates that we saw during the pandemic, but even we're seeing rates below pre-pandemic levels. And I know it's not just in influenza, but this will have an impact on how successful a pandemic rollout will be. I'd like to move to the last slide, please. So this is, it says CSL Seqirus a proud champion. This was our press release. We've been working with BARDA. This was an announcement to say they had asked us to fill finish antigen that we already had made for them over the last sort of 18-month period that we have clinical studies ongoing on. And we were absolutely thrilled to get this award to be able to fill finish this vaccine so that it can be ready to be deployed in the US. And I think I'm finished now, and I'm going to hand on to the next speaker from Sanofi.
Thank you very much. Our next presenter is Jillian Ritchie from Sanofi. Jillian, your slides are up. You have the floor.
Thank you, Dr. Hopkins. Can you hear me okay?
You are loud and clear.
Excellent. Well, good morning, everyone. Thank you for the opportunity to address the committee and preparation capabilities of our approach to pandemic preparedness. I'm Julian Ritchie, and I head up our public affairs patient advocacy activities here in the US. And if we go to the next slide, I'd like to outline a couple of areas that, in addition to the broad portfolio of vaccines you may be familiar with Sanofi across pediatric and adult vaccines, that pandemic has been, and influenza has been, a cornerstone for more than 70 years. We've been engaged in the research, development, and manufacturing of flu vaccines. And that includes both our domestic capabilities, and these were demonstrated during 2009 in our H1N1 response, where we were able to bring product to market in under 3 months. And it extends to also our established networks and capabilities that we continuously explore new technologies and processes that can improve response capabilities, much in line with what you heard of the goals that Dr. Oshansky mentioned from BARDA in terms of speed and quality of response. And lastly, I think very much in line with what Lorna just shared, although we're focused on the production aspects of the pandemic preparation, we really have to take a holistic approach to innovation and think about that all the way through implementation. And so we'll touch on that in a moment as well, because as we know, a vaccine doesn't prevent disease until it becomes a vaccination. And so a key part of any pandemic response has to be that infrastructure that's able to deliver the vaccine through supply chain and readiness, all the way through provider readiness. And I couldn't agree with what Lorna mentioned earlier, that this is somewhat like fixing a hole in the roof. You don't want to do it when it's raining, you want to do it when it's sunny. And so the opportunity to make more robust our delivery systems and improve confidence in vaccines is key in order to truly have a complete response in the case of a pandemic. Next slide. So Sanofi's influenza footprint for R&D and production is an expanding network that's global in scope. As you see here, you have an outline of multiple facilities around the world. And specifically in the US, you can see a number of markers to indicate the number of facilities and the breadth of the coverage, the diversity of the platforms here, ranging from our Swiftwater, Pennsylvania site, where you see egg-based, recombinant, and our fill finish facilities, up to Waltham, Massachusetts, where you see our mRNA R&D Center of Excellence, to Pearl River for recombinant, bulk, and Ridgefield for fill finish. So it's a very robust program here in the US specifically. And all of this has a complement and redundancy throughout the world in order to provide large volume quickly. Next slide. Several points have been mentioned about surveillance, and Sanofi works closely with many government agencies in order to have early detection and early intelligence on activities that are happening throughout the world. Our detection mechanisms help determine us what is circulating, as you can imagine, as well as when a response is warranted, when it reaches that threshold, and where. And so working with global partners such as WHO, HERA, and others, we engage with networks such as the Global Influenza Hospital Surveillance Network and the newer ALIAS network to better ascertain information for both our internal planning and decision-making, as well as to share with government partners. While official cues come from government agencies, the early indicators and information that we get from systems like these help us scenario plan and evaluate potential response strategies ranging from prototyping to at-risk manufacturing and determination of how pandemic response activities could impact manufacture of other routine vaccines so that we balance the response and provide that information for open decision making. Next slide. As you've seen before, the partnership is key, and this is a team sport. We've mentioned government partners. Our US-based production capacity has grown substantially over time via our partnership with BARDA. Working together, strategic investments in manufacturing facilities, in supply procurement, all of these have ensured that we have capacity that can be kept warm throughout the use in seasonal influenza and constantly ready as capacity that can be redirected and prioritized to support a pandemic response. Some specific points in this timeline include starting back in 2004, first working with BARDA to secure year-round egg supplies and ensuring that those were secured flocks that have expanded over time in order to support the robust egg-based cornerstone of the pandemic response program. But this has expanded as technology and knowledge and science has improved. And in 2009, BARDA investment in recombinant technology began and has grown to include pre-pandemic stockpiles and expanded recombinant production here in the US. This volume, together with our other manufacturing capacities for Northern and Southern Hemisphere I showed you on the previous slide with our other facilities, mean that more than 250 million people per year are immunized around the world for influenza. Next slide. So as you saw on that map earlier, our facilities do support production via multiple platforms. On the left-hand side here, ranging from the reliable and proven platform of egg production, moving across to the right to our production of recombinant technologies and ongoing development work with mRNA, we explore these new technologies while continuing to innovate and refine processes in our existing platforms. Oftentimes we focus on the new technologies in terms of the conversation, but it's important to realize there's continuous innovation that's happening even with the existing platforms. You see here listed some of the components that currently are in the toolbox and regarding H5 and H1. These are constantly watched and iterated in order to understand how response can be improved while engaging with new technologies to meet the goals that Dr. Sansky mentioned earlier in terms of faster response, but all the while it has to be a quality response. We want to make sure that anything we bring forward is going to be able to improve not only on speed, but also on the quality, the efficacy, and the performance that will in fact protect people and inspire confidence in vaccination. Next slide. And finally, in the spirit of our discussion of production response approaches, I'd like to share an approach that Sanofi is investing in regards to a new format of production we call EVF, or evolutive vaccine facilities. A challenge when you look back at our existing technologies and where we've been is that traditional facilities are more fixed in nature and their construction, time, and expense to change them is often substantial and a very significant rate limiting factor for any response. The EVF concept is bringing in the modular production approach that allows rapid change from one process to another through standardized forward-looking processes, all of which are compliant to regulatory standards and can be rapidly moved and integrated as needed, which is especially important in the dynamic nature of a pandemic assessment and response. What's really interesting about these facilities is through their design is it is possible to produce multiple vaccines and or biologic products, even those based on different technologies. So we talk a lot about mRNA, insect cell, mammalian, et cetera. You can do this and be able to do this in a manner that provides investigational scale production as well as commercial scale production. So we look at this as being the future of manufacturing. It also enables us to incorporate state-of-the-art digital designs. Cobots, an interesting term for the idea of robotic assistance that's able to assist workers in their work, as well as full artificial intelligence for processes improvements. Single-use processing, being able to use pieces that are going to ensure better compliance and quality assurance. We mentioned that they're flexible and more easily expanded because they can be running concomitantly with different technologies or can be deployed against single technology for greater volume. And ultimately, with the newer construction and the nature of the design, they can support environmental improvements in terms of neutral footprint, lower energy consumption, and goals towards zero emissions. So in summary, we're committed at Sanofi to innovative manufacturing and technological solutions that address the existing needs, because many of these things are based in that warm concept of supporting during routine and seasonal administrations, to the emerging issues that happen and the responses needed in order to be on the front line for a pandemic response, in order to make sure that we're bringing the best quality products as rapidly as possible in order to meet patient and public health needs. So with that, I thank you and welcome questions.
Thank you, Julian. I want to thank all the members of the panel. Are there any questions or comments from the members of the committee, either in the room or online? Molly?
Yes, I was curious for our speakers, should there need to be a vaccination response to the circulating H5N1 in birds or cattle? Should we need to vaccinate humans? What impact would that have on the availability of seasonal influenza vaccine? I guess I'm asking if both pandemic vaccine and seasonal vaccine can be made at the same time.
I'm happy to offer perspective on that. So that's absolutely one of the key decision-making factors that we bring forward in the consideration. So many of the points I mentioned in terms of the planning are looking at what impact does this have on manufacture of other products. And many of these facilities are built in a manner where we can do this in a complementary fashion. But that's part of the close collaboration we have with our government partners in order to determine the priority and the timing of what's necessary. And so fortunately, our flu production is very far progressed. So this year, it wouldn't have that kind of impact. But it's also something we have to watch very closely in order to ensure anywhere in the process, from early stage down through fill finish, that's taken into careful consideration.
I would also reiterate what Julian has said, and it's all to do with dialogue with BARDA on when we have at the moment, the manufacturing window to produce more H5N1 or H5N8 is there because our seasonal production has finished. But if the demand was such that it was during seasonal production, then that decision would be made by BARDA and probably based on a WHO pandemic call.
Thank you. Next question is from Mitch Rotholz.
Thank you all for the presentation. Question for the group is, during the COVID pandemic, one of the limiting factors was the packaging for the vaccine, so the actual syringe or vial supply. How are you planning for that in your production facilities for when you would need to step up production? Do you have that already lined up, or strategy of either pre-filled syringes versus the vials? Give us some perspective on what you're thinking there.
Maybe I'll take that first and then hand to Julian and Christine. So we stockpile based on our contractual volumes, either the vials or the syringes. Obviously vials, because you can have more doses in the vials. And what I can say during the COVID pandemic is that, say, from our Liverpool facility, we actually made those vials available to for a competitor vaccine because it was a new vaccine that was in development. And in Australia, we were able to use the vials that we had for the pandemic response, the pandemic influenza response, for our COVID response. So we always keep the vials or the syringes as per our contractual needs.
I can complement that and build on. Lorna, you've hit it very clearly. The volume from a supply chain is always a consideration. I think what also was substantially different in COVID was the recognition of fragility of supply chains in regards to the depth of our suppliers. And so I've seen substantial increases in redundancies for different components of process in order to make sure we have secondary suppliers, and are thinking more robustly about making sure that we do have the depth of supplies that we talk about here. And those conversations have advanced through the conversations with our government partners in order to make sure that's part of the contractual considerations. But it's also a key part of our own manufacturing for other routine products as well. So knock on wood, it's all about the preparation, and it's hard to make any guarantee, but I think the awareness has increased dramatically so that we are all more aware of supply chain as well as national border issues and thoughts around that, so that we can all be much more well positioned for anything that we should face in the future.
Next question is from Steve Rendernacht.
Hey, again, thank you much for those presentations. One of the keys to this is obviously rapid response, and these new platforms that are much faster, more efficient, are the key in the direction to go. But they also seem to be detrimental to the confidence from a patient standpoint, because that speed too often was relayed as cutting corners and not doing things appropriately in some way. And to reinforce that, any tips that you folks can share with me to share that, hey, there's no corners being cut, and the same routines are being done on this. But I hear that over and over. Thank you.
I agree. I hear that a lot from friends, family, as well as people, physician friends asking. And I do try and explain that studies were done at risk in parallel, whereas as an industry, we would wait and see the results of one study before going into another study in terms of cost, not in terms of safety, but also that because a lot of the documentation is prepared in advance, one is able to respond quicker. But I think it's probably an answer that needs to come from government, from the regulatory bodies, rather than from industry, because obviously when it comes from industry, it has that ability to be tainted. So I think probably it needs to come from the regulators to explain how the process was speeded up. And most of it was because we took, parallel investment choices rather than sequential investment choices.
I could add to that too. One of my hats is in regards to patient advocacy. And so confidence in vaccines is a key concern. And I was very pleased to hear Lorna mention that, as well as you heard my mention of that holistic approach that we need to take. And very often, those of us who are deep inside of this focus on the front end, the innovation piece, being able to take that through to confidence and make sure that our messages are clear. I agree that oftentimes we're not the best groups to convey this, that our government partners, but it really needs to be all of us being able to support together the harmonized message. And I feel like what we saw during the pandemic is that some of the better uptake and confidence came from people understanding that steps weren't skipped, timelines were compressed. And so emphasis on quality, it's something I realized as I was even sitting here listening to our presentations that I don't think I emphasized enough, that all of this about speed and technology is all a function of what we can do to convey that there's quality and proof that anything new is better than what we had in the past. And making sure that we convey that, I do think, is an essential component, because otherwise we'll do a tremendous job of responding to a pandemic or other need with product, but not with immunization. And that's not success.
Hey, thank you.
And I would add to that, I think fora like this, where we can get the message out that there are prior preparations in place, that we're already doing things now so that we have tools available more rapidly, may also help to reinforce the messages that we're trying to do things so that we are better ready without cutting any channels or cutting any of our efforts at safety and effectiveness. I want to thank all the members of the panel. We are now going to take a brief break. We're going to step away until 10:45 a.m. Eastern Time. Thank you for joining us for our June 2024 NVAC meeting.
Thank you.
Produced by the U.S. Department of Health and Human Services.